Perfect. So let's dive into it. Yeah, so for those of you that haven't heard of BioRender before, we're an online tool that helps scientists create professional figures in minutes. So thanks for joining BioRender 101. Today we'll be doing a quick intro about BioRender and also a demo of our tool as well. So if you do have any questions here for us during the webinar, feel free to select all panelists and attendees if you have any questions, and we'll be sure to get to them as soon as possible. Alrighty. So yeah, thanks for joining. Today, as mentioned, we'll go over a brief introduction and a BioRender demo. Before we do get started, I'd like to launch a quick poll just to understand who we have in our room today. So feel free to fill it out now. Really looking forward to seeing who we have joining us today. Okay, awesome, great. So I'll keep going. I'll leave that poll up for a few minutes to let you, I'll check it out. There are 2 questions, so once you finish your first question, please scroll down and answer the second. So brief introductions about ourselves today. I'm joined today by my colleague Francesca Kim. My name is Philip Brook. I'm on the business development team here at BioRender. I'm really looking forward to showing you what BioRender is all about. I'll let Francesca introduce herself.
Hi everyone, this is Francesca, and I am currently on the science communication side at BioRender. Really looking forward to taking you through the demo today. Thank you, Phil.
No problem. Thanks, Francesca. So I'm taking a look at the poll. It seems almost everybody has filled it out. We're going to go ahead and end that poll, share the results. Seems like we have a really good mix of people today, so we'll definitely try and get to everything today. And if you do have any questions, please don't hesitate to ask us. It seems we have some people that have not signed up for BioRender yet and some that haven't used it but have signed up. So not to worry, we'll definitely show you how everything works today. So prior to being BioRender as we are today, we were actually a custom medical illustration studio called Anatomize Studios. So Anatomize was run by our CEO, Shiz Aoki, and her professional career led her to become the lead scientific illustrator at National Geographic for over a decade. She regularly created many beautiful scientific visuals, kind of like this one you see here on the screen. So on the side, she also worked extensively with scientists and with popular academic journals such as Science and Nature. So she identified that most scientists and labs simply don't have the budget, resources, or connections to collaborate with professional medical illustrators to visually communicate their science. So these researchers would end up using PowerPoint or something similar, spending hours and sometimes days to make an illustration. This in turn led everyone coming up with dozens of different ways to show essentially the same pathways or reactions or methods with different degrees of success. Shiz then asked the key question: what would happen if we somehow made all the graphic elements involved in scientific figures standardized and available to all scientists? So this was actually the seed of how BioRender started. So as you can see here on the left-hand side, these are some figures that we found online, showing pretty much the same thing, and this is it on the right created in BioRender. So this is just a quick speed through of the BioRender platform. As you can see here, it's really easy to use. Simply drag and drop icons from the left into your figure, but we'll definitely get into the demo today and you'll be able to kind of ask as many questions as you'd like. So just a quick list of some of our use cases we find. Obviously, publication figures and presentations, grant applications. The list really goes on and on. Pretty much anything you can create a scientific figure for, you can use BioRender for. So our goal as a company is to give every scientist the tools and the practical knowledge to visually communicate their research. If anyone has any questions, please ask in the chat And I'm going to go ahead and hand it over now to Francesca to show you what BioRender is all about.
Thank you, Phil. Do you mind allowing screen share, and I can go ahead and share my screen and take everyone through the app?
Of course.
Let's see over here. I hope you can see my screen okay. It should be just on the BioRender home page right now. And let me know also if my audio quality is okay. I can adjust it accordingly. Great, seems like it looks good and sounds good. So I'm gonna go ahead and get started. From the poll, it actually looks like a lot of you joining us today haven't had a chance to use the platform yet. So I'll go into the basics, and then we'll work our way up to the advanced features. So hopefully everyone can get a little bit something from the tool. So if you haven't had a chance to sign up, you can go ahead and click on sign up free using this button. Feel free to do so. It takes less than a minute. You can follow along to the demo as well. I already have an account, so I'm just going to sign in using this button right here. And just like that, it's going to kind of auto log me into the BioRender homepage. And if you haven't used it before, your homepage might look a little different than mine, and don't worry about that. As you build up your figures over here, you'll start building up your personal library. So these are all the images I've made in the past, or used in the past actually, to create my figures. Over here, there are 3 different types of galleries. So right now I'm under My Illustrations. Everything here is private to me. You can kind of toggle over and create teams on BioRender as well. So I know a lot of you working in a lab setting are probably working on projects together with either your PI or other graduate students. In industry, you probably have a project team. So feel free to go ahead and create a team on BioRender, and it'll make it really easy to share images and collaborate on these figures together. So how you can do that is you can actually go right here, clicking on your name on the top right-hand corner. You can go under account. And then if you just select team, you can invite members through their email address. You can feel free to also remove members if you'd like. So I know Cindy, my colleague Cindy's on the line. I can just invite her really quickly. Cindy at BioRender.com. I can send her an invite. There she is. And if I wanted to remove her, I can also go ahead and remove her. So that's kind of how you administrate teams on BioRender. You can set a name. Mine says BioRender, but obviously yours will say, Smith Lab or whatever makes sense for you. And then once you create a team, you can really easily share images. So I'm going to go click on this 3-circle dot over here. And I'm actually going to go right here to share. I'm going to click on this button. I can toggle between send to email. So if you want to send it to someone directly, for example, Cindy at BioRender.com, or if you want to share it with the team that you just made over here, you can just go ahead and click on send. And just like that, you might see that some of my images over here have this little icon on the top left corner with a team icon. And that just means that this image has been shared to the team gallery. Everyone on my team can now access the image and use it as well. And any edits that I make to this image will be reflected in the team gallery for my team to see as well. So yeah, that's basically how you share images. And then this little paper airplane icon on the top left corner basically just means that someone has directly sent an image with me or shared it with me in that first way that we just saw it. So yeah, that's basically the gist of how your illustrations work. And then your team gallery. You can also create folders on BioRender. So if you're working on like a publication for Nature, maybe your first one, you can go ahead and save that. And then all you have to do is kind of click and drag these images right into the folder. Like so. And then you can also drag these folders around to orient and organize yourself. I hope that makes sense. Let me know if you have any questions in the chat, and I'm happy to address anything or, answer all your questions as we go. Phil, you can feel free to also unmute yourself and ask any questions that are asked very frequently. Other than that, I won't go too much into, the nitty gritty, but you can sort by last modified, or the name of your image. So if anyone has been online shopping before, you know what that's about. And then you can kind of go lowest to highest, or highest to lowest is what this little arrow does. So yeah, feel free to go ahead and play around with your gallery and see how you like it organized. And once you set it up, you can go ahead and start creating an image, which is, I feel like the most exciting part about BioRender. And there's actually two ways to get started. So the first way is to start from a brand new illustration by clicking on this blue button right here. And the second way is actually to start from a pre-rendered template. So I'm going to go ahead and click on this template and show you how you can utilize that, and we can come back to it at a later time. But over here, basically, if you toggle, what I did was I toggled between this gallery, and then I'm just clicking on templates now. I'm toggling here. And what this brings me to is like a page of, pre-rendered and pre-designed templates across every field of life science. We have hundreds and hundreds of different science templates available for you. They are all fully editable. So if you can go ahead and click on any of them, you can see the description of the template, who we collaborated with on. And if you want to know more about it, you can go ahead and search for, further readings. A lot of them have references available for you. So this one is based on this one article, etc. So go ahead and search for it. There's a lot. So I would recommend, going through either categories or searching directly. I see in the chat here, it says Parva is actually needs icons for lower back pain. So in terms of templates, I might, try typing in anatomy, and you can go ahead and see that we have a lot of different anatomy pictures. Maybe you want, an image of a person and highlighting different areas. You can go ahead and search for that. I know we do have a lot of icons related to it, but this is basically background images. Yeah, so feel free to go ahead and search. If you search up pain, we have a lot of different icons related to pain, or templates related to pain as well. A lot of lab protocols and timelines as well. So I know this is super useful for really visualizing that complicated process. If you're a grad student trying to explain, to your PI what you just did in the lab, or, you're doing any sort of presentation outside your lab and you're trying to explain the very specific mechanisms, I feel like images are really, really great for that. If you are a lab manager and you want to make these protocols, or lab tech, we want to show how you can use these different types of equipment. Images are very powerful for making that manual or lab protocol for your team. And yeah, as I said, all of these are fully editable. So if I want to go ahead and click on one of them, I can go ahead and use the template by just clicking on use template right there. And just like that, I'm kind of brought to my canvas view. I really like the layout of this one because it goes from, 1 to 6. They're all sectioned off for me and kind of listed, but I don't personally work with a C sponge, so I can go ahead and delete any aspect of it. Maybe I work instead with the liver. I can go ahead and type in liver. We have different types of icons related to liver, as you can see. Anything from a fibrotic liver to a regressed fibrotic liver to maybe a polycystic liver or cirrhosis. I'll pull this one out for now, maybe this fibrotic liver, and adjust it. I can send this to the back, like so. And you can see how even if this initial template didn't fully relate to the research I'm doing, I can really easily use it as a starting point to start building out my figures. So because we're working with the liver, maybe I want to bring in some hepatocytes here. We have a lot of different color options. So if you just click on it, click on the color that you want, and then just drag out the thing that you like and resize it like so. I'm just copying this over. One really quick design tip as well, if you have multiple cells and you want to convey that message that there's a lot, kind of randomize it. The smaller ones you can fade into the background as well. So yeah, just a cool little design tip right there if I want to knock back the opacity of some of them. So it looks like there's a lot. You can go ahead and do that. And then obviously I'm not working with symbiont cells, so I can change any of the text. So the idea is that all of these kind of templates, you can use it as a starting point or even a final point and edit them as you wish. Any questions on how to use templates, feel free to throw them in the chat box and I'm happy to answer that. Otherwise, I'm going to go back to my gallery by clicking right here on this button. And you can see that I didn't actually have to press save or save my image at any point. It kind of auto saves right here. This is the image I just made. And then if you go and click on this 3 circle, whoops, I misclicked right there. But if you go ahead and click on that, right here, you can go into version history and you can actually retrieve previous versions of this image. Over here, it kind of auto saves every change or every minute. So you can go back in history. If you don't like a change that you made to one of your figures, you can go ahead and retrieve that. Similar to Google Docs if you know about that feature. Awesome. So that was BioRender templates. We'll come back to that in a bit, but feel free to take a look and try searching for, you know, your area of research or what you're really interested in. And we actually have a full team working full-time every single day to update these templates. So you can check back every week to find new ones weekly. I'm going to go ahead and start from a brand new illustration now. So this is the kind of second way you can start from an illustration. I can click on this blue button, can make a title and the size, but this is, you can always change this later on, so you don't have to worry too much about picking the perfect size and name. And just like that, we're kind of brought to what we like to call the canvas view on BioRender. I'm going to hide the slides feature for now, but this is just the beta feature we're testing out. And if you imagine yourself kind of just sitting at your desk staring at a blank piece of paper, the analogy that I like to make is that this white square is basically like your paper. So anything that you include in the white square will be part of your final image. This gray area is like your desk space. I really like to use it for reference images or icons that I use really frequently. I can drag things out here, but anything that bleeds outside of this white space won't be part of your final figure. So feel free to use this as your creative board and then just kind of the white square as your final picture, basically. A lot of you are probably working off laptops. So if you want to save more screen space, you can just kind of toggle this out. And then before we get started with the figure, I just want to quickly orient you to how you can navigate yourself around the canvas. So over here, we have the zoom bar. You can go really zoom really far in or really far out, depending on what your needs are. We have this ruler that you can toggle on if you click on this button. If you can see over here, it's kind of turning the ruler on and off. And this just allows you, these are personal reference lines. So these are by no means part of your final image, but if you are working on, say, a publication figure, those multi-panel figures always look more professional when they're fully aligned. Borders, you should keep very consistent as well. So these are just reference images that or lines that you can make for yourself and kind of section out your figures. I'm going to turn it off for now, but just know that that's available for you. And then also this grid view, if you want to work with a grid, you can increase the size of the grid or decrease it. That's also available for you. I'm going to turn it off for now, but those two options are right here. And then on the bottom right-hand corner is kind of like the help desk. So if you've used Facebook Messenger or WhatsApp or any kind of instant messaging platform, you know, AOL or MSN back in the day, it's a very similar concept where you can go ahead and click on send us a message and chat instantly with any of our CX representatives, our customer support staff, and they usually respond in less than 2 minutes, so it's very quick. I will say though, the live chat hours are right now from 9 a.m. to 6 p.m. Eastern time when our representatives are online. So if you ask during that time, it'll be very short. Otherwise, you can also reach out to this email, which is support@biorender.com. To reach our support team, and they'll be able to get back to you within 24 hours, or usually I think like 1 or 2 hours. But yeah, feel free to go ahead and send them a message if you need any help at all with your figures. We do have our FAQ guide here as well. So all our features should be highlighted. For example, if you want to learn about crop, we have these really cool guides that help you learn. They all have really good animations that actually highlight how to use every feature. There's design tips in there as well. So very helpful. We've been revamping it. So hopefully that'll be helpful for you, which you can access right here. And then throughout today's session, you might see me use some sort of keyboard shortcuts really quickly just to make it faster. And don't worry, they're all listed over here. So if you want to follow along and you're curious about what kind of shortcuts I'm using, feel free to keep this open on the side. It might look a little different depending on if you're Mac or PC. I'm on a PC right now, so it says Control and Shift, but it might be Option for you if you're on a Mac, and that'll reflect right here in keyboard shortcuts. Awesome. So I'm going to go ahead and get started with the figure. I'm going to recheck the polls right now, and it seems like a lot of people in the call right now are kind of in the molecular and cell biology space. A few of you are also in, neuroscience and behavioral science. If you didn't get a chance to input your field into the poll, feel free to just throw it into the chat box, and I really want to make today's session as relevant for you as possible. So yeah, go ahead and throw in your fields or anything you want to see, and I'm happy to highlight those as well. And yeah, all right, let's get started. So because a lot of it is cell biology and immunology, I'm going to go ahead and go into my icons. So BioRender, we have what we call icons, which make up parts of your figure. So if you've used things like Lego, if you remember playing with Lego as a kid, the icons are basically like the individual pieces which make up your final structure. And then, yeah, you put these icons together and you get your final figure. So it seems like there's a lot of people in immunology, so you can go ahead and look up cells. We have over 40,000 icons in our library. So why don't I just go ahead and type in T cell, search for that. We have a lot of different icons related to T cell. I'm going to use this kind of generic one, so I'm just going to click on it. Let's see, what color do I want to use? I'll use this one for now, and I'll drag that out. I can resize it and just place it there. I can also rotate it around. Maybe I'll do like a T cell and a cancer cell interacting, so I'll go ahead and type in cancer. I like this tumor cell, so I'm going to go ahead and drag that out. A lot of you are in cell biology and immunology, so maybe this will be quite relevant over here. Yeah, and just like that, I have, my T cell, my cancer cell. I want them to interact, so maybe I'll go ahead and throw in CAR T cell receptors. So I'll go really specific, and I'll drag that out, kind of resize it, and reformat it here. And one thing I want to note is that these are what we call icons, but we actually have 3 different types of icons in the app. So we have this really simple one, really quick and easy to use. You just kind of click and drag it out, resize it like you just saw me do. Our second type of icon, you might have noticed some of them have purple boxes on the bottom left corner. So what that is if I drag that one out, this is actually a grouped icon. So you can see between this icon and this icon, they both look very similar. Some may say that they're almost identical, but the big difference is that you can see that this one has a blue bounding box. You can change the color, the size, and the orientation, but can't really do much about editing individual parts of this. Whereas if you see any icons that are surrounded by a purple bounding box, like this one or this one or this one, it says double click to edit group, so you can always go ahead and double click on it. All of these you can kind of resize and reformat. If you drag around any of these like little bullseye looking points on any of our shapes, it kind of changes the shape. So feel free to go ahead and play with that too. Maybe you made a mutation and you deleted this domain of the CAR T cell receptor. Go ahead and delete that. All these lines are editable as well. So all our icons do have different degrees of editability. And if you see that it is a grouped icon, I would fully recommend just double clicking on it and seeing what that does. We have a lot of different types of it. So if you have like a mouse, for example, with a tumor maybe, You can go ahead and, even change the size of the tumor if it's, regressed, or if it's early stage, late stage, after treatment, before. You can go ahead and do that. If you're working in the genetics area, so I see that a few of you are kind of in that space as well. You can type in, Drosophila and all of these grouped icons as well. So this one has a mutation on the wing. The head, you can change the eye color or even the whole head color over here. Any of these wings, maybe this one's not the right one, and you kind of have a different wing right here that you want to put. Maybe there's just a mutation in this one wing. Any of these are all like, you can take them apart and edit them individually. So yeah, feel free to go ahead and take a look at that. Yeah, I hope that's not too confusing. So far we covered normal icons and grouped icons, just to recap. And grouped icons are the ones with the purple box around it, which you can edit further. I'll delete that for now. Maybe I'll put a receptor on the cancer cell as well. So I'm going to just type in receptor and see what we get. So we have anything from very specific receptors, even closed channels, open channels, to very generic ones over here. I'll just use this kind of generic circle-shaped receptor, and we now know that it's a grouped icon, so we can double click on it to edit the colors. I personally like to unify my colors with what I'm trying to convey. So for example, this one is in particular, I want to put on my cancer cells. So I like to put my antagonists, or quote unquote the bad guys in my story, as more of a reddish color, because I think subconsciously that's just how our brain kind of registers things. And then our quote unquote good guys, I like to kind of unify in cool tones, because it's just the nature of how we tell stories in media. So yeah, I have my cancer cell and my T cell over here. I can go ahead and type in some T cell receptors and add them on for all you immunologists out there. I apologize if this isn't fully correct, but I do believe this might be true. And then MHC class 1, I believe, was on the cancer cell. So I'm gonna place this right on here. So you can see how I didn't actually have to know how to draw any elements, as long as I understand the science and I know what kind of story I'm trying to convey. It's just like playing Lego with scientific images. You just kind of have to place everything together and put it the way you need it to be. Awesome. So over here, I have my T cell and my cancer cell. I'll throw in some DNA. So I'll go ahead and type in DNA. Which kind of brings me to my third type of icon. You might notice that some of these have this blue paintbrush on the bottom left-hand corner. And what that does is I'll just drag it out. These are actually super cool. I was really excited when we launched this, because these are very dynamic icons in which you can kind of drag out the different points. And when I let go, the DNA will automatically recalculate. I can pull these like blue nodes around to reshape it. For example, if I want a DNA that's very squiggly, I can go ahead and do that. Yeah, so that's just kind of what we like to call our bio brushes on BioRender. Over here, I can go ahead and change the color and have this very dynamic DNA. This is actually a really cool tool. So why don't I highlight it a little further? I'll group all of this together. I'll put it back into my workspace. Like I said, we can put images right here. And I'm just gonna go to, from right now we're at icons, I'm gonna go down to this brushes button right here, as you can see on the left-hand panel. And what I'm gonna do is I'm gonna select any of these kind of linear brushes and drag it out. All of them work very similarly in that when you drag out these nodes, it won't actually skew the icon. So a traditional icon, for example, if I take a cell, this cell, and I drag out this, it'll just get longer this way. You can see here that if I drag this one out, it doesn't skew the icon. It actually just adds more cells over here. And if I let go, they'll all recalculate so that they're perfectly aligned. So that's what the brushes does. It's really, really cool. You can drag out these points and kind of make different nodes over here. Maybe you want to make kind of like the cell cleaving. You can do that. And we have them available for a lot of different things. So if you're working with, blood vessels, we have those endothelial cells that you can use to demonstrate like cross sections. If you're working with a gut microbiome or anything related to, enterocytes, you can go ahead and drag that out, and you can see if I let go, all of these cells will automatically recalculate so that they're all perfectly aligned. I don't know if you've tried making this on Illustrator or PowerPoint before, but I know it's notoriously hard to make all these non-leaky, tight junctioned kind of cell barrier line here. We also have DNA, anything from single-stranded DNA, where you can make these cool replication forks, like so. And then vessels as well. So yeah, feel free to go around, play around with it. We also have the phospholipid bilayer. I know I don't know if you've tried making like using circles and sticks to draw this on a platform before, but this, is really hard to normally do. And we've made it so that you can very easily create these bends in the membrane. Maybe you want to make an endocytosing membrane. You can just drag out these points. And every time I drag out this white node, it turns into a blue node and it becomes an anchor point. And then another one appears. So you can kind of infinitely keep doing that. You can double click to get rid of them as well. Awesome. So that's just kind of how our bio brushes work. Another cool thing I did want to highlight is that if you did want to edit any parts of this, so for example, maybe you're working with the disease state of the gut over here, and you want to convey that there's something going on, I can go ahead and actually convert this into a grouped icon. So what I'm going to do is go into brush options. I'm going to separate this into editable icons, like so. And this actually now became that second type of icon that we talked about earlier. So if you recall all these mouse icons and CAR T cell icons. So this was our first type of icon, just to recap. This was our second type of icon, which allowed you to go ahead and double click on it to edit. Our third type of icon were these kind of dynamic brushes that we can go ahead and change around over here. And this third type of icon can actually be transformed into that second type, which now I can go ahead and double click on it. We can make these cells diseased. So I can go ahead and click on these cells and make them really inflamed, or the villi are kind of breaking apart. I can even delete any of them to make it slightly leaky. Yep, you get the idea. So yeah, I hope that's not too confusing. 3 types of icons, 1, 2, 3, and then 3 can transform into 2. Let me know if you have any questions. I know we're covering a lot of content. Happy to go through anything slower or faster as well. So feel free to kind of ask your questions in the chat box.
Hi, Francesca. We have a question here. Someone wanted to see if you could kind of draw your own shapes and icons. Could you show how that works?
Yeah, that's a really great question. So we can actually draw our shapes and icons. There's 2 ways where you can do that. So if you want to go ahead and go into insert line, we have this kind of custom pen tool that you can go ahead and draw any shape. One really cool thing is that if it's a line and you let go, you can have these nodes, and I don't know if your hand's really steady. I know mine's not the steadiest, so I can edit them further afterwards. Another really cool trick is that if you do have these like custom shapes and you close it, and if you close the line, it becomes a shape. So then you can fill it and it becomes its own shape. And like a little blob, you can use it really great for like proteins if you're trying to draw Cas9 or anything like that. Our shapes as well. So if you go into shapes, all of these are fully customizable. So like I said, if you see these like bullseye points, you can drag them around and see what they do. So this one makes it kind of narrower, and then this one adds more points, etc. So our shapes are very dynamic. I would fully recommend that you go ahead and use it. Maybe this arrow, you want to add a gradient. You can go to fill color, toggle over to the gradient and then add those as well. And you can even drag them out to adjust, how much of that gradient. So if you can see the blue over here, if I drag it out this way, it'll be more towards the blue. If I drag it out this way, it'll be more green, and so forth. I hope that answered your question about drawing your own shapes. Another way what you can do is you can also kind of upload your own figures, but I'll get to that very shortly. Awesome. So I'm just going to go ahead and try to finish this off right here. I have my two cells interacting. I want to convey that there's some sort of signaling pathway going from this CAR T cell receptor to the DNA. So why don't I go ahead and insert a line over here? And our arrows are very dynamic. I can actually click on it. Very easy to use. And just like the bio brushes, these blue nodes you can drag them around to adjust it. And I can kind of infinitely make any shape that I need to over here, like so. And if you're not happy with it, you've added too many nodes, all you have to do is double click on it and it'll go back to deleting that node basically. So, we have these right angle signaling arrows. Maybe I want to add one over here to convey that the signaling is causing some sort of cascade towards the tumor cells. I'm going to go ahead and do that. I can color match all my arrows. So why don't I go ahead and zoom into this one arrow and show you what we can do. I can change the width of it to make it really thick or really thin. Keep it at a 3 for now. Can make it dashed if I needed to and kind of adjust how big that is over here. I can create the kind of line, fade, or gradient by just clicking on this button. Can change the head to like maybe an inhibitor or a neuronal synapse. I'll highlight actually our neuron arrows in a bit. They're really, really cool. And I know there's quite a few neuroscientists in the room with us today. So highlight that in a bit as well. But yeah, you can adjust kind of the head size over here, make it really big or really small, like so, and then line type, opacity, all that stuff. And as you can see, every time you kind of click on a different feature, your top toolbar changes depending on what you can do with that. So yeah, if I click on arrows, I get all these different options. Why don't I go ahead and change the color and match it? Like I said before, I really like matching all my elements to what I'm doing. I like this one, so I'm gonna go ahead and do that. Maybe I'll change this one as well and kind of pick this color over here. And then last but not least, I'll add in some cytokines. That's kind of going towards the tumor cell because the T cell is probably attacking the tumor cell. I'll rotate it and kind of resize it over here. Awesome. So we have the beginnings to our figure. All we have to do is add in labels now. So I can go ahead and insert text. I'll add in T cell. I'll make it bold, and I like to match this as well. So any labels as well, I like to match it with the element because anyone outside of your field might not really be familiar with, what a T cell looks like, or if you get more specific, what certain receptors look like. So I always find that kind of making that uniform color really helps your brain register it, just like 1 or 2 seconds faster so they can pay attention to more of your presentation and your research rather than trying to re-navigate, yourself to like, oh, which one's a T cell, which one's a tumor cell, etc. And another quick design tip is for any action items that aren't labels. So for example, this one is signaling, but signaling obviously wouldn't be like a full out label. I like to put it in like kind of a different font. So I like to italicize it or make it slightly different so that it's clear that this is not a label of this arrow. It's just an action that's going on here. Awesome. So we have, our mouse here. Maybe and then we have our tumor and T cell here. I'm going to actually group it, and I might come back to this in a bit. Maybe I want to use it in the future. So I'm going to actually add it to my favorites by clicking on this star button right here. You can also right click and add it to your favorites. And what that does is it actually builds out your own personal library of favorite icons. So if you've used Spotify or Apple Music before, or even made a mixtape back in the day to kind of make a playlist for maybe working out, you kind of get the sentiment of like you don't want to be searching for the same icons or the same songs over and over again. You kind of want to have, all your favorite tunes available. It's a very similar idea here where you want to have all your favorite icons available for you. And then you can just pull that out really quickly. So for example, in the future, maybe I'm working with this tumor mouse and I want to convey that there's something going on, in here. I can just go ahead and make that really big, like so. Maybe like this. And then I can maybe in the future add in my image that I just made because I've saved it. So I just don't have to remake it over and over again. So the more you kind of build out your own personal library of icons, the faster it does become to create your image really quickly. One other thing is you can also adjust your canvas size if it no longer makes sense with what you're trying to make. Why don't I make it at least actually a little bigger over here? And then now I have my mouse and my tumor T cell interacting right here. And one other thing is that you remember the teams that we created in the beginning of the session? You can actually share this image with them as well. So for example, if I've made this, I've invited Cindy to my team and I feel like she could use this image as well. I can go ahead and click on this blue share with team button on the top left-hand panel or the top left-hand corner of all your icons. And what you can do is you can just click on update. And this is all personal to you. So this, your favorites is private to you. But if you toggle over to the team tab right here, this is all actually private to your team. So now Cindy can go in and go ahead and use that figure for her presentation. So really great for kind of unifying concepts. I know a lot of times in lab meetings, if every single person is representing their T cell in a different way, maybe you're using a different type of cell or a color, it takes a little bit more time for your whole team to kind of re-navigate themselves around your image. So it takes that much longer for you to actually communicate your research because you're trying to discuss your results, but people are still trying to understand the background and like what the T cell is. So great idea. I found that team efficiency goes up a lot once you start standardizing these type of items. Also great for collaborating if you're both working on a figure for your publication or your posters, you can go ahead and use that. Why don't I finish this off by just adding in a pathway? So I'm going to go ahead and type in pathway. I'm going to actually toggle over to templates, and we have a lot of different types of pathways here. So I can select one. Why don't I actually go ahead and use a blank pathway? Just so I don't offend anyone if the science is wrong. Yeah, and over here you can kind of go in, search for the template that is relevant for you, and you can add that to the illustration. And if you add something to the illustration, it just kind of drags that as a grouped icon so that you can go ahead and place that. So this is actually a really great way. Another kind of design tip is that when you're conveying or you're trying to communicate your research, I would always go from macro to micro. So for example, if someone just showed you this image by itself and it's not really part of your immediate research field, you probably won't understand what it's trying to convey. It'll probably take you a little longer. But if you are working at a molecular or cellular level and you also showed this kind of macro image from the organism to the cell to the molecular level, it's always a better communication tool that you can use. Like so, and then now you fully know that this is a T cell. This is like activation inhibition going on with this interaction right here. So yeah, highly recommend at all times to kind of utilize that to your advantage of showing both macro and micro views. Yeah, and then why don't I just add in an arrow really quickly to show that this is highlighting this over here. And last but not least, I'll go right into our uploads. And we can, upload any kind of data sets. You might be working on GraphPad or Prism. You might be, on Pymol looking at proteins. Go ahead and upload files directly. This is an image of a blood smear. Maybe that's one of your microscope results. You can upload that and include that in your figure. Maybe you're part of a biotech and you want to include your logo or your lab logo. You can go ahead and upload that. Oh, this is really big, so I'm going to zoom out actually. And the zoom, what I'm doing is I'm just holding control on my keyboard and using my mouse scroll. And then when you're really zoomed in, a really cool shortcut that I use often is spacebar. So if you hold spacebar, you can like pan around without having to zoom in and out too much. So I'm going to go ahead and add in this little watermark or whatever logo I want. I can knock it back to the background to make it an opacity. Yeah, and that's basically the features of BioRender. I do want to highlight though one really cool feature called our PDB, which is the protein data bank. So we are actually directly integrated with them. I can show you how to use that, but right now I have I'll add in this and then for here, add in your data set. And maybe one more microscope image right here. All your results for your figure. Like so. And I want to kind of highlight this, maybe this is my ligand of interest and I really want to highlight that, or any proteins that you work with in particular. We are actually directly integrated with the PDB. So this is the last tool over here on the left-hand corner. And if you know your alphanumeric code, I'm sure if you work with the protein, it's kind of, you know it like the back of your hand. But if anyone is working with a protein in particular, feel free to throw it into the chat box. I can try looking it up and we can pull it up together. Otherwise, you can use a generic one, maybe insulin. But yeah, if you know a particular protein, feel free to let me know and I can look that up for you right now. This is a crystal structure of Drosophila kind of insulin over here, and the alphanumeric code for this crystal structure in particular is 6FEY. So if I go into the protein data bank, I type in 6FEY, you can load it and you can see that this is the correct one. It's the insect insulin over here. You can view it in a 3D space. So I know if you want like a really quick way to visualize your protein, it's a really good way. You can like zoom in and zoom out over here. Like so, you can convert it to a van der Waals view as well if you want to. Yeah, super cool feature. It's really fun to play around with as well. And if you want to see it by secondary structure or hydrophobicity or, by molecule, you can go ahead and toggle between the different options. You can change the design a little bit. And it's just a quick way to kind of visualize that protein. You can save it as a new icon and you have that available for you now. Another really cool thing is also, if you want, you can pull up the exact same protein kind of with its cartoon view, because I know a lot of times protein visualization, the structure and the secondary structures and the surface structures are all very important. So what I like to do a lot of times is also drag the second icon out and knock back the opacity for one of them and kind of overlay it so you can see both the surface view of the protein and then the secondary view of the protein, group it together and kind of have that available for you. Awesome. So that's how the PDB works. Obviously, if you work with this a lot, you should add it to your favorites. You don't have to do it over and over again, and then you can just drag it out in the future. Maybe you want to, include this here. Whatever you want, you can kind of do that. And it looks like we have our figure now made. So what I'm going to do is I'm going to go ahead and export it if you're happy with it. I'm going to click on export right here in the top right hand corner. And before I do that, actually, I'm going to check it in grayscale. So this button right here, I can check it in grayscale, make sure all my contrast is okay. A lot of times, if your contrast isn't great, you can go ahead and see this kind of animation shows you how to improve it. Checking it in grayscale really helps for your colorblind audience. Also, if you're submitting a grant or publishing, a lot of reviewers will print your grants or papers, or even your readers might kind of print it in black and white, so you can double check to see that all your figures are good to go in grayscale by using this button right here. And then once you're happy with it, go ahead and export it. I would always recommend PNG for slides, but if you are working with a lot of microscope images, very photo heavy figures, JPEG is kind of your best bet. And then if you have a lot of text, I would recommend PDF. But for now, I'm going to do the PNG. I'm going to knock back that background by clicking on transparent background. For presentations, 150 dpi is the way to go, just because it's a really high resolution, but it doesn't kind of sacrifice the loading time. For print, I would recommend 300 just because it's a very high resolution. And obviously if you're printing it, you don't have to load it. If you're using it for a commercial website, I would recommend 72 just because it loads a lot faster. It won't slow down your presentation or page. So I'm going to go ahead and export that, and it's just going to save directly into my thing over here. And yeah, just like that, I kind of have it saved. If in the future, this is like a new Google slide image, I can go ahead and drag that out, and then this is the image that I just created. I hope that makes sense. Let me know if you have any questions, and I'm happy to answer them further now. Yeah, so those are pretty much all the features I wanted to highlight. If you understand all of it, you can, fully make your first figure, and I have confidence that you'll be able to. A few other features I wanted to highlight, just because there's a lot of neuroscientists on the call with us today, is our neural kind of synapses and structures. So we have a lot of different icons related to the brain. And we actually have these really cool neuron lines over here. So if you go under insert line and click on neuron, you can make these pathways really, really quickly in a matter of seconds. Any kind of pathways like this. This just kind of flips the tail and the head back and forth if you drew it the wrong way. Maybe you want to convey that this is, you made some sort of manipulation here. This is an active site of some sort. Or you created a lesion. You can use these custom blobs to kind of highlight different areas. Like so. You can even knock back the opacity, change your color presets, and etc. So yeah, really, really cool tool to quickly create these structures. I know this has taken me a long time in the past, so hopefully that's helpful for you. And anyone working with cycles as well, we have these cyclical kind of arrows right here where you can make this into a full 360 circle. You can cut it at any point and kind of make that, if you're working with genes, those tools get a lot easier with this feature. Why don't I go ahead and knock back the head size like so. And maybe you want to make a gene map, like a plasmid map, and this is the promoter, so you can get rid of the head here. You can change the line colors. Anything you want, you can pretty much do it here. like so. Why don't I make this line width a lot smaller? And then I can just make these plasmid maps really easily. Yeah, I hope that was helpful. I also want to be mindful of everyone's time. So unfortunately, we are kind of coming to an end, but happy to answer any last minute questions in the chat. I think, wow, there's a lot of questions that came in, and I think Cindy and Phil did a really good job answering all of them. So thank you, Cindy and Phil. Is there anything else that I missed at all, or anything that anyone wanted to see? I'm happy to highlight any last minute features right now. Otherwise, I think we've covered everything. So yeah, let me know if you have any questions. And I guess we can call it a day here. And I hope everyone has a wonderful day. Hope the session today was helpful. And yeah, have a great rest of your week.